through biology.
Ready?
Sure. Do you know, when I was about 8 years old, it was my dream to go to UCLA and learn their planetary geology degree.
At 8.
Yeah.
Wow.
I was weird. Never got to
Bring
UCLA.
home a lot of rocks when you're Bring home a lot of rocks when you're
No. I was one of those kids who brought home the stick bugs and all these horrible things, like lizards and bugs and stuff.
Was the officer yet?
Yes.
Yes.
Well, you can see it's
Dream
project.
squishy, was it crushed, like made it crushed normal
What's that? Fired up, let's go. Okay.
Back then I
Stop on Mars, you need to spend like 10 years training.
So lots of interest in Mars. There's been interest in Mars since telescopes were limited, and they described there being canals there, and the idea of Martians, and certainly NASA is one of its primary targets, and the idea of follow the water. One of the things we'll talk about today is the idea: could we be Martians? Right? Is it scientifically reasonable? I might argue some of us are. If you look at early Mars, you've got a planet that's smaller than Earth, would have cooled quicker than Earth, would have condensed water. We've got evidence for liquid water on Earth at 4.2 billion years. It would have been there quicker. If you argue that late heavy bombardment on Earth sterilized the planet, we've got life emerging within, tens of millions of years. So there's plenty of time on early Mars for there to be liquid water, for there to be an atmosphere, and for life to potentially have launched. So what would have happened to an early Mars, right? And we take kind of the view, we've got a red planet, we've got water. The major water activity died out after that late heavy bombardment, right? But if we had kind of flowing and standing water and stromatolites similar to what we had on Earth during late heavy bombardment, was it purely a sterilization, or could material have been launched off of Mars and landed on Earth? We got meteorites that have made that trip. So if material was launched from early Mars and landed on Earth, could we be Martians? So here's our early Mars, taking the kind of the current terrain, and they just threw in a shallow sea. Could have been saline, right? Whether or not there would have been oxygenic photosynthesis or not, or whether it would have been anaerobic. Is this red?
brownie.
Brownie.
So,
Okay,
yeah.
so they're not quite at. Can't tell. We're not quite at the oxidized planet stage yet. But this as a target, because it's smaller, it's easier to launch stuff off of Mars than it is to Earth. Because Earth is larger, it provides a greater gravity sink to what's there. Now, when you talk to geophysics types that like to blow up things, and I work with Eileen Ryan at New Mexico Tech. They have an energetic materials research testing center, and they blow up things. Tanks, planes, you name it, they've blown it up at this testing center. We brought rocks, right? And one of the things that she explained to me is that you don't necessarily have to just blast it so hard that something will fly off, but there's a spalling that goes on when the shock wave goes down and rebounds, that will fling stuff off. So we said, let's hit rocks with that kind of force and see what happens to bacteria. So we took a sandstone, not heavily consolidated, but reasonably so, and it had about 10 to the 5, 10 to the 6 bacteria per gram. And so the first impact here, okay, you can see between 3 and 4 milliseconds, there's nothing happening there, and then there's an impact, there's a flash, and we're starting to produce a crater. And literally within that rock, we had about a 4-centimeter crater. that was dug out of that surface. We picked up the sand that came off the bottom, and then we looked at bacteria that were in the crater versus bacteria that were farther away versus control rocks. And basically the counts were the same, right? We couldn't really distinguish. So, but this isn't the kind of force you would need to, you know, launch something off the planet. This was just, let's just give the rock a hit, put a shock wave through it and see kind of what happens. So you ramp up the force. A rock, 5. There's literally a flash on impact. Okay? And because of the shock wave, the rock exploded. Right? So just the shock waves going through the rock kind of disintegrated. We had some chunks, you can see chunks kind of flying off that are up here. We picked up the sand, we picked up the chunks, and counts were the same. So the planet, and this is the kind of force that you would need to spall something off of a planet. So the next step was, let's hit it with a sterilizing impact. Okay, so great big steel projectile. By the way, these things are traveling at kilometers per second. So not kind of meteorite fast, which is tens of kilometers per second, but for something that's kind of high density and high mass, certainly very quick. So when we hit it with the steel impact, we didn't see the flash. We saw it going out the back of the rock. And we were hard pressed to find anything bigger than 2 pieces of sand stuck together. And the bacterial counts went up.
Is that just because there's no surface area?
What's that?
Is that just because there's no surface area after it's all broken apart?
So, exactly. So if you look at it, we saw, you know, on the surface of the rock, microcolonies of bacteria. Right? So if you've got microcolonies of things like this, your standard or your conventional, you know, you take that and you go for, you know, kind of colony forming units per gram kind of thing. That microcolony, you know, will produce 1 colony forming unit. And the only explanation we have for the bacterial counts going up is that if it shattered so much that it literally ripped apart microcolonies from the surface, then you'll increase the count. So that the shock wave that just literally destroyed the rock didn't affect the bacteria. They're like, sure, it's fine.
Was it cultured immediately after the impact?
It was cultured. No, they were brought back to Western. So we weren't able. We're in the New Mexico desert. So we left.
So how many days or weeks?
They're put on ice and probably processed within the next few weeks. They're all maintained at 4 degrees. The rocks themselves have been collected, you know, kind of months before, and it's Navajo sandstone. It's millions of years old. They really don't. There's not a lot of food in there for them to grow. Yeah, so the energy that it takes to launch something off the planet will not kill bacteria. We followed it up by putting kind of endospores in ice and looked at kind of an impact. We vaporized the ice, still didn't kill the bacteria. Right, so they can withstand the kinds of forces.
What about when it's coming into us?
So usually what happens within a meteorite is that if it's friable, it can explode in case you have a shock wave there. Otherwise, you're going to produce a fusion crust. So you will literally melt the outside of a rock, but the inside stays cool. Okay, so and we've looked at kind of the Tegish Lake meteorite and things like that have come in. So the stuff gets to the surface fairly intact. It can still have organic carbon. So carbonaceous chondrites deliver organic carbon and phosphorus and all sorts of nutrients right to the surface.
So while it's possible, would it be likely though that rocks from Mars will make it all the way to Earth, like be in the proper trajectory here?
Yeah, so the last one we'll talk about is the Allen Hills meteorite, right? So there's a whole suite of Martian meteorites within collections that are there, so they can make the trip, right? A lot of assumptions here, right? Mars had life, launched off, got to Earth. You know, interstellar travel, you need to travel within a rock to be protected from solar radiation, right? If you're on the surface, there's going to be sterilization going on. Okay? But if you're traveling within a rock, then it's possible. So we go to Mars itself, and the rovers, they've been targeting iron-rich regions. So in Meridian and Planum, the spectroscopy says it's full of iron. They were this kind of ellipse is, on the scale of kind of, 10 kilometers by 50 kilometers. That was the landing zone. Basically bring it there, blow up big balloons and let it bounce around until it stops and unfold and take out the rotor and see what they see. When they got there, first outcrop that's ever been seen on Mars. Geologists, everything's always just been chunks and debris and, kind of dust storms blowing stuff around, and they found outcrop. And I was looking at this, and you look at pictures and stuff that they provide, you really need a scale bar, right? Because I saw that, and it's just like, wow, look at this. You've got, there's some topography on Mars, right? Not as spectacular as Australia, but there's some topography there. And when the rover drove up to it, it was like, oh
no.
It's the size of the table, right? That was the outcrop that they had, but you got rocks that are the first evidence of the balloons and where it was kind of bouncing around. The nice thing about this is that they were able to drive right up onto it to take pictures, to analyze, to grind into the surface. What they found is that they had layered strata, okay? And embedded within the layered strata were hematite nodules, right? We see this kind of stuff on Earth. The spectroscopy, the Mossbauer, which targets iron, gave spectra that are reminiscent of iron oxide. It's like, well, it is Mars, right? So it's not a big leap there, but that it was pointing towards jarosite, right? We found jarosite in the supergene copper system. We found jarosite in Rio Tinto. Okay, so anytime you're in acid-generating environments or acid mine drainage environments, you get jarosite. So it's speaking to weathering of a parent material, producing an acid sulfate mineral that we know happens under surface wet conditions on Earth. So if we look at, flashback to Morenci, we can pull out our iron and sulfur oxidizing bacteria, and kind of the yellowy material that's there, that's classically jarosite. And it's kind of the yellow boy. We've got our iron oxide, we've got our copper sulfate, and we've got jarosite forming, with the help of bacteria. Kind of the classic environment where I've seen jarosite is Rio Tinto. Right? We've got 100 kilometers of pH 2 to pH 3, absolutely covered in iron oxide. And what we found at this site that was interesting was that if you look down literally on the cobbles themselves, you've got such a fast dehydration. Right, in the rock surface, you get there in summer is worse. It hurts. Right? You touch the surface of the rock and it's above 60 degrees Celsius. Right, so you've got a very quick dehydration going from the freshly hydrated things that are full of bacteria to hematite. So all you have to do here, we get our fresh oxides, you strip out the water, it just evaporates, and you're kind of consolidating and sealing and trapping what's ever there. So if you think of the Mars system, would the hematite nodules or those types of things be good candidates? What are you going to sample, right? We're talking about that. What do you cache? They're talking about, it's a little kind of big there, kind of a pop can sized container of rocks to bring back from Mars. It'd be about $1 billion, right? Very valuable material. But what are you going to put in that? And how many samples would you collect to put in that? We know from looking at the iron oxides that are fresh in this system, it's loaded with filamentous bacteria, right? We don't have fresh oxides though on Mars, right? And we can go into the fresh oxides from Rio Tinto and we can culture out filamentous iron oxidizing bacteria, right? And the whole argument there is, is there the hope of some sort of extant life or is it all extinct, right? And the likelihood is that, certainly from a surface perspective, it's likely extinct, right? The other thing about Mars is that even though it's far from the sun, there's photolysis of water that goes on, and Mars is too small to hold on to hydrogen, and the hydrogen is being lost, right? Just the gravity isn't enough to hold on to it. What that does is it enriches in oxygen, so the surface of the planet has things like hydrogen peroxide, right? We use this kind of a sterilizing agent. Aerobes can break it down with catalase or oxidase. And we have the perchlorics that are there as well. So we have these oxidizing compounds that, you know, limit the likelihood of finding stuff near the surface. Now, once they went up over that outcrop and had a look around, they actually found that it was just much, it wasn't just kind of a table scale, that they're looking at hundreds of meters, if not kilometers, of all of this sedimentary material. And when they look at the surface of it, the jarosite, the finer grain material, is more susceptible to weathering. And what it did was literally the surface was covered with hematite nodules. And these are, they describe them as blueberries. It's like, I have no idea why. You know, you're looking at kind of millimeter scale sorts of materials. And we can find hematite nodules in riverbed iron on Earth and all sorts of sandstone environments where they weather out. I've looked at hematite nodules from Australia. These are some larger ones from Utah. They just weather out of the sandstone. They're just lying there in the ground, just like is going on at Mars.
What percentage of those good?
What's that?
What percentage iron are those good?
These things can be very hot, so they're almost pure hematite. It's like the kind of this Kanga thing that we're talking about, right? So if you're getting to, you know, you're mostly that, then kind of banded iron formation, you're maybe kind of 30–40% iron, and this thing can get up to, you know, kind of 66–68% iron. So very, very rich from an iron perspective. The problem with these in the Kanga is that it has the phosphate, right? So it's not necessarily a good resource because of that.
What location is this picture from?
This is from Utah. So just near the Four Corners region, a little north of that. Arches National Park in that area. So it's, yeah, we've got these flat horizons, and in depressions you get kind of water and endolithic bacteria and things like that, but these are kind of weathering out of that environment. The ones we've looked at from Australia, very reminiscent of the ooids, right, in the stromatolite or the thrombolite that we've had. That we've got, you know, kind of some sort of core material that's more kind of sedimentary or a nucleation zone, and then we've got laminated structures. Did not really find much evidence of fossil bacteria out in this lamination. So it's this link between, is there a biology and abiotic realm? Looking at the material here, it's like, oh, really hard to, you know, I wouldn't bet my career on something like this, right? You know, we've got filamentous-like structures, but some of them seem kind of cut in half, although a lot of them are hollow, right, which would suggest a template, right? But it's not, you know, it's not just an obvious fossil, right, that you think, okay, look at this, it's absolutely full of bacteria that you can see stuff kind of growing and dividing. Now this stuff here is 3 billion years old, right? So a lot can happen to it in that time. So this whole idea of preserving things over geologic time, the older it is, the more suspect you are that something has happened to that material. The other thing that was interesting about the kind of the rover cruising around Mars was when it got its wheel jammed. So they had some dust kind of caught in the wheel, it jammed, and they were driving along, dragging the wheel. And what it did was it exposed salts. And when they started kind of digging around and driving around and, you know, doing donuts and, you know, kind of disturbing the surface, and this whole idea, and from the spectroscopy, there have been certain areas that are kind of patchy and white and very reflective. But this idea of sulfate salts, right? That if you've got something crashing out with sulfate, right? Whether it's calcium, magnesium, you know, maybe iron to a certain degree, but these evaporites, right, tend to be quite bright. And the idea, again, pointing to, you know, to make these things, at least at some point, there had to have been liquid water. That's the idea that the planet was much more active than it certainly is today. We can go to vast lakes in British Columbia, Canada. Okay, so this is bank sulfate, just like you would pull off the shelf if you're growing sulfate reducers or something like that. If you look up, you put up Handbook in Chemistry and Physics, you can dissolve 700 grams per liter. Right, Epsom salts. Right, so just absolutely spectacular environment, and all the colors that you got here. So this is ultramafic rock, there's groundwater, there's an acid source kind of up gradient, and as it's weathering through this material, it's dissolving, the sulfate's coming from the pyrite, it's neutralizing, it's coming out at about, I think this one was about pH 10, so very alkaline environment. And the red that you've got here are halophilic bacteria. Okay, things that can withstand that. So we went in, there's a whole series of these lakes that are there, evaporated ponds. The students who are out there are kind of grabbing samples. When they finished today, their feet looked like raisins. It was just kind of scary, right? Because it's that salty, it's pulling water out of their feet. And they couldn't, the ones that tried to go out there with shoes or boots, they would just get stuck. And then they're trying to dig the boots and shoes kind of out of the system. In this pond here in particular, they were pulling biofilm out, and the biofilm was full of salt crystals. Like, well, the evaporites make sense, right? But they found a whole bunch of salt below the surface of the water. So whether it was old evaporite that had just sunk, we thought, it's hilarious, the microbial mats are making salts, right? Wouldn't that be great? If you take the salts, you can take the salt crystals that are from here, you put it in the microscope, look for fluorescence. Photosynthetic organisms have nice fluorescent pigments, and a lot of the halophilic bacteria can harness light energy, get bacteriorhodopsin, and we could find bacteria trapped within the salt. So you go back to Mars and this idea of wet making salt. Would salt be a good sample? That as the evaporation, that if there is organisms growing in the water, and this is hypersaline, right, then would that be a good sample to kind of bring back? We did the spectroscopy, so looking at kind of salts, we could find, kind of band numbers or wave numbers that correspond to organics. There's salt signatures in there, but we've got carboxyls and carbonyls and amino-functional groups. So using spectroscopy to target biological material, and we could differentiate the biological material from abiotic salts that we made in the laboratory. We could enrich cultures, so we could pull out sulfate-producing bacteria and cyanobacteria, and when we did that kind of thing, we had spectacular spectra, and we're looking at thresholds. How many bacteria do you need? And it was about 10 to the 5, 10 to the 6 bacteria per gram to be able to see it using spectroscopy. Right? So would spectroscopy then be a good tool that as you're picking up sulfate or salt samples to analyze it and go, can we detect anything of promise in that? Yes, collect it, or no, pitch it out and grab another one. That you want to be able to triage samples as well as it could. And the remarkable thing that happened within these cultures is that we got salts forming. Right? In fact, the controls stayed completely liquid, and when the bacteria were growing there, the salt crystals grow completely in the test tubes.
Cool.
So what do you need for a salt to precipitate? If you picture this magnesium sulfate that's dissolved, right?
You need there to be more of the salt in the solution than you have, than the
So we
solution.
didn't have more salt. The abiotic tubes, you look at them, and they were clear, and they were just salty. Right. So the idea of nucleation, okay, that's a good one. So within this, nucleation is definitely part of it, and this was kind of basically a clear salt water. So what else happened? So in terms of here, to keep the magnesium and the sulfate, right, from interacting with each other, right, to make the idea of a solid, you've got, in terms of magnesium, if you've got oxygen, right, interacting with the magnesium from the water, right, then it can't interact. Likewise, you can have the hydrogen from water binding to the sulfate. So you gotta have, there's layers of hydration that keep stuff in solution, right? And if you disrupt those layers of hydration, then things will crash out, all right? So if you think of the bacteria, organics have layers of hydration, right? The bacteria are trying to hold on to the water, that they're gonna change the water activity. In terms of the bacteria themselves, pulling water into their cells, or just even the idea of water interacting at the atomic, molecular level, right? There's layers of hydration that go on. One of the ways you can purify different organic components, we did it to separate blood components, is you add in salt, right? The salt takes the water from the organics and the organics precipitate. So you can salt fractionate of different organics. Well, what I think is happening here is that it's the reverse, that the organics are stripping water from this system and then causing the salt to precipitate and trapping the bacteria at the same time. The Allen Hills meteorite, this is the one kind of created all the controversy. So what they found within the meteorite were these little globules. And the globules are made of carbonate. And they've got pyrite. And there was kind of a movement, was active here at UQ. It was active in a group in Texas, and it was this idea of nanobacteria, right? And, you know, basically what you've got here is something that's on the scale of 20 nanometers in diameter, you know, times maybe 70 nanometers. So could that be a cell? Exactly. Right? Put your phospholipid membrane onto that. Never mind peptidoglycan or an outer membrane or any sort of wall to protect you from exploding. But you look at, you know, your phospholipid membrane. You got 7.5 nanometers just with that. You know, need a membrane on the other side too. So you got your membrane here, and then you've lost another 7.5 nanometers. So in this case, you got 5 nanometers inside. Can't fit a ribosome. Ribosome's 20 nanometers.
And that's the smallest thing you got in that.
So it's just far too small, right, to hold this stuff to make life, right? But this is, you know, published in Science. What's that? So could there be a relic? So the idea of could there be nanofossils? Right? You look at bacterial flagella or fimbriae, right? We talked about this idea of nanowires and things like that. You could have a flagella that's, you know, kind of 10 to 20 nanometers in diameter, and could be microns long. So could it be a remnant or a piece? And it's like, sure, right? But it's just far too small to be bacteria, right? We can make carbonates, calcite, in cultures of sulfate-reducing bacteria while we're making pyrite, right? That's why these globules are so intriguing, right? How do you make carbonate? You can describe photosynthesis. You can say we'll give it protein-rich material in the